Hello everyone. In this class, we'll study about splanchnic and skeletal muscle circulation. Splanchnic circulation. Splanchnic circulation means circulation of blood through abdominal viscera. It has three components: circulation through the gastrointestinal tract proper and the mesenteric attachment, that is intestinal circulation. Second is circulation through the liver, that is hepatic circulation, and third is circulation through the spleen, that is splenic circulation. Intestinal circulation. The major function of the intestine is digestion and absorption of nutrients. The normal intestinal blood flow is about 20% of the cardiac output at rest, which increases about 50% following a large meal. Without this increase in blood flow, proper digestion and absorption do not occur. Blood supply. The gastrointestinal tract is supplied by three main arteries: celiac, superior mesenteric, and inferior mesenteric arteries. Superior mesenteric artery is the largest branch of the aorta that carries more than 10% of the cardiac output and supplies many parts of the small and large intestine. The branches of mesenteric arteries, that are called small mesenteric arteries, form an extensive vascular network in the submucosa of gastrointestinal tract. The branches from these arteries penetrate deep into the muscle layers and form the arterioles. The arterioles supply the tip of the villi. Intestinal mucosa receives about 60 to 70% of the total intestinal blood flow. So this diagram shows the blood supply to the intestine. Venous drainage. The capillaries in the villi drain into the venules that finally drain into mucosal venules and then to the veins. Direction of blood flow in the arterioles and venules in a villus is opposite to each other, which forms a typical countercurrent exchange system. This permits diffusion of oxygen from arterioles to venules directly. Therefore, supply of oxygen to the mucosal cells at the tip of the villus is reduced. Normal gastric blood flow is around 40 ml per 100 gram of the tissue per minute. Intestinal blood flow is around 60 ml and pancreatic blood flow is around 80 ml. Regulation of intestinal circulation. Neural regulation. Sympathetic stimulation causes constriction of the mesenteric arteries and arterioles, thereby reducing blood flow. These effects are mediated via alpha receptors. During exercise, the sympathetic vasoconstriction shifts blood from mesenteric vascular bed to the skeletal muscle. Autoregulation by metabolic and myogenic mechanisms. Metabolic regulation occurs mainly by adenosine, osmolarity, and the potassium. Adenosine, it causes vasodilatation and it is going to increase the blood flow. Increased osmolarity of the blood passing through the intestine as occurs following ingestion causes vasodilatation. And potassium also causes vasodilatation. Regulation by the GI activity. GI blood flow increases with increase in the GI activity as occurs during digestion of the food. Increased absorption of the food also affects the intestinal blood flow. Several products of digestion like chyme, glucose, fatty acids are potent vasodilators. Functional hyperemia. Gastrointestinal blood flow increases by food intake, which is partly due to the secretion of gastrointestinal hormones like gastrin, cholecystokinin, and partly the products of digestion. Increased blood flow helps in digestion and absorption of food, which is called as functional hyperemia. Hepatic circulation. Liver is an important visceral organ as it performs major metabolic activities of the body. Liver requires adequate blood supply to carry out its routine functions. Hence, the blood flow is more to liver, and hepatic blood flow constitutes around or about 28% of the cardiac output. The blood flow to the liver is derived from two sources. One is portal vein and another one is hepatic artery. Portal vein provides about 75% of the blood flow to the liver, as the blood has already passed through the capillary bed of GI tract oxygen has already been extracted once the oxygen concentration is less here. The hepatic artery provides 25% of the total blood to the liver. It is fully saturated with oxygen. Therefore, oxygen utilized by the liver is primarily derived from the hepatic arterial blood. Normal blood flow is 58 ml per 100 gram per minute, and it is about 28% of the cardiac output. So this diagram shows the hepatic circulation where it gets arterial and blood from the portal vein. The portal vein is formed by the venules of the intestine, and the splenic circulation and the gastric circulation. Whereas through the hepatic artery, it is going to receive oxygenated blood. Hepatic arterial regulation of the hepatic blood flow. Hepatic arterial buffer response. Alteration in hepatic arterial blood flow occurs reciprocally with the portal venous blood flow. It compensates for change in the portal blood flow. Neural regulation. Both hepatic artery and portal vein are innervated by sympathetic vasoconstrictor fibers. And metabolic regulation is also seen in hepatic blood flow. With increased hepatic activity, the accumulated metabolites like carbon dioxide and hydrogen ions cause vasodilatation and increase hepatic arterial blood flow. There can be autoregulation. The portal venous system does not autoregulate its blood flow. However, autoregulation exists for hepatic arterial blood flow. And it is also controlled by regulation of the intestinal activity. The activities of the GI tract increase the blood flow to the liver via portal system. Clinical importance. Liver filters the blood and removes the toxic substances before allowing it to the general circulation. The nutrients absorbed from GI tract pass through the liver and are stored or metabolized according to the need of the body. When central venous pressure is elevated, as occurs in congestive heart failure, the hydrostatic pressure in the vessels of the liver increases. This results in exudation of fluid into liver tissue and causes tender hepatomegaly. This is an important feature of heart failure. Fibrosis of the liver increases hepatic vascular resistance, which increases portal venous pressure, which consequently increases capillary hydrostatic pressure in the splanchnic vascular bed. This results in transduction of the fluid into the abdominal cavity that causes ascites. In portal hypertension, esophageal varices occur, which may rupture and bleed to cause hematemesis. To prevent this, portocaval shunt is performed to decrease the portal venous pressure. During hemorrhagic shock, about 60% of the blood from the liver can be expelled into systemic circulation to increase the blood volume, which is achieved by constriction of the capacitant vessels of liver by the sympathetic stimulation. Hence, liver is an important reservoir of blood in humans. Coming to the splanchnic circulation, it is supplied by the splanchnic artery, and it is regulated by the sympathetic fibers. Splenic artery is supplied by the sympathetic vasoconstrictor fibers. Sympathetic stimulation diverts the blood from the splenic circulation to the hepatic or systemic circulation. The sympathetic stimulation causes vasoconstriction and contraction of the smooth muscle present in capsule and trabeculae of the spleen. Splenic contraction releases adequate amount of the blood into the general circulation, which serves as a protective mechanism in exercise and shock. Skeletal muscle circulation. Skeletal muscles constitute largest mass of the body. In adult weighing 70 kg, the weight of the skeletal muscle is about 30 kg, that is total 40% of the body weight, whereas the blood supplies only about 15% of the cardiac output. However, resistance offered by the blood vessels of skeletal muscle accounts for 25% of the systemic vascular resistance. Therefore, caliber of skeletal muscle blood vessels contributes significantly to the control of blood pressure. Moreover, circulation of blood through the skeletal muscles depends greatly on the activity of the muscle. Normal blood flow at rest is around 2 to 6 ml per 100 gram per minute, and during exercise it increases 20 to 50 times. The blood supply to the skeletal muscle is derived from the skeletal muscle arteries. One of the important features of this circulation is the presence of large number of arteriovenous thoroughfare channels. These blood vessels short circuit the blood from the arterial side to the venous side by bypassing the capillaries. They are innervated by sympathetic fibers, which are of two types: sympathetic vasodilator system, sympathetic vasoconstrictor system. Sympathetic fibers supplying the blood vessels of the skeletal muscles are cholinergic. Therefore, stimulation of these fibers results in vasodilatation in sympathetic vasodilator system. Whereas sympathetic vasoconstrictor system, there are no sympathetic noradrenergic fibers that secrete noradrenaline and cause vasoconstriction. Okay, so here you have noradrenergic fibers that leads to vasoconstriction. Regulation of the blood flow, neural regulation. At rest, resistance vessels exhibit a basal tone that occurs due to continuous low frequency activity of the sympathetic vasoconstrictor system. This tonic activity is greatly influenced by activity of the baroreceptor reflex. Stimulation of the baroreceptors results in dilatation of the blood vessels of skeletal muscles, and decreased carotid sinus pressure causes carotid sinus pressure causes vasoconstriction. The muscle resistance vessels contribute significantly to maintenance of blood pressure, as blood vessels in the skeletal muscles constitute the largest vascular bed in the body. During exercise, at the beginning of the exercise, the stimulation of sympathetic cholinergic fibers increases blood flow to the skeletal muscles due to vasodilatation. But once exercise continues, the metabolites accumulate in the exercising muscle and cause vasodilatation. The metabolites and local hypoxia cause dilatation of the precapillary sphincters and increase blood flow through capillaries. This increases oxygen and nutrient supply to the skeletal muscle. Local regulation. In active muscles, blood flow is regulated by metabolic factors. ADP, adenosine, hydrogen ion, carbon dioxide, lactic acid, and hypoxia they cause vasodilatation and increase blood flow. Increased temperature in the active muscles during exercise also contributes to vasodilatation. Regulation by some of the physical factors. During exercise, the contraction of the skeletal muscle mechanically compresses the blood vessels, therefore decreases the blood flow. Thus, during active muscle contraction, the blood flow is minimal, and during strong sustained contraction, blood flow almost ceases temporarily. However, in intermittent contractions, blood flow occurs during the relaxation period, and venous walls prevent backflow of the blood in the veins between the contraction. Clinical importance. Skeletal vascular bed is largest vascular bed in the body. Therefore, the tone of resistance vessels of the skeletal muscle contributes significantly to the maintenance of systemic blood pressure. The blood present in the skeletal vascular bed contributes significantly to the venous return, that is the skeletal muscle pump activity. When the skeletal muscle pump activity increases, venous return increases, and therefore cardiac output increases. When venous walls become incompetent, especially in the superficial leg veins as seen in pregnancy or in old age, the vein becomes dilated and tortuous. These are called as varicose veins. Cardiac output decreases in such conditions due to stasis of blood in dilated and incompetent veins. These conditions are treated by injection of sclerosing agents or by using the elastic stockings. This is all about the splanchnic, hepatic, and skeletal muscle circulation. Thank you.